She actually presented this research at our Nebraska Academy of Science meeting that we just had last month on her the characterization of tardigrade microbiomes.
Thank you for the introduction. Hi, once again, my name is Mary Morris, and I attend College of St. Mary. And today, I'm going to talk to you guys about the characterization of the tardigrade microbiome. First off, we have a few objectives for this research. The primary focus of the study was to determine the most effective method for microbiota extraction and DNA purification from within the tardigrades to determine the microbiome characterization. And then our second kind of topic is the resiliency of these microorganisms and help shape the way that we develop new treatments and therapies for humans in regards to their protective abilities. So first, quick outline. We're going to talk about tardigrades, why they matter, what they are. Second, we're going to talk about microbiomes and what they are and why they matter. Third, microbiome isolation. Fourth, limitations we've had with our research so far. 5, current and future studies, and then finally questions following. So first, like I said, tardigrades. This is a front view and then a bottom view of the tardigrade. They're pretty cute and they're microscopic. They have 5 different segments on their bodies. They have a tube-shaped mouth from the front to the back of them. And they have internal stilettos or teeth that allow them to grasp onto their tube. And they have 4 pairs of legs on 4 parts of those segments. And they usually have 3 to 4 claws per leg depending on the species. And then they don't breathe through their proboscis. They actually do gas exchange through the body's surface. So tardigrades are extremely resilient animals, and this is why we're so interested in them. So whenever they are induced with stress, they tend to enter a cryptobiosis of sorts, which is known as an extreme anhydrobiosis, and this is also known as their ton state. They can survive up to 25 years in a lab with an extreme lack of water in this ton state. They can survive temperatures of up to down to negative 260 degrees Celsius and up to 150 degrees Celsius. They can withstand pressures up to 6 times that of the human body can withstand. And then radiation up to 10 times what the human body can withstand. And then they are also one of the only creatures we know to this date that can survive being in space. So where do we find them? We find them primarily in lichens. There's 3 different types of lichens we can find them in: crustose, foliose, and fruticose. All of them are very similar, just differing in their shape and where they're found, primarily on trees and fences. Here are 2 pictures that a former student of ours, Elizabeth White, took during her research over tardigrades. On our left, we have a more clear, larger one. And on our right, we have a smaller, more solid colored one. These are just 2 different species that we found in the Omaha area. So now, microbiomes. So a microbiome is the full collection of genes of all the microbes in a community found within or on a separate organism. It consists of over 100 trillion bacteria, archaea, fungi, protists, and viruses, specifically for the human body. The human microbiome is all of our microbes genes, and then can be considered a counterpart to the human genome, which is all of our human genes. And so here's a quick graph to kind of show that, how important the microbiome is to the human body. In blue, you can see all of the microbial cells kind of shown, and then how many human cells we have, just a comparison. And then this is a quick breakdown of all the different types of microbes that are found in the microbiome on the human. In blue here we have bacteria, which is one of the largest ones. And so how does this relate to the tardigrade microbiome? Well, there hasn't been a ton of research done to figure out how prevalent the microbiome is for the tardigrade. But here we can see that this is the tardigrade's specific DNA. And then these are the foreign or not already determined DNA that we find located on or within the tardigrade. And we know that these specifically foreign DNAs are bacterial, so that would mean it consists of the microbiome. But there's still 20.3% in this study that we don't know whether or not they belong to the microbiome or to the tardigrade itself. So that's pretty interesting. And the DNA can be cross-referenced with other DNA libraries. So the reason why we don't know what this is specifically is because it hasn't been actually categorized yet. So just to further deepen the importance of the tardigrade's microbiomes and how it relates to the human microbiome, in this specific study, we could see how D-sub proteins paired in their DNA protects their DNA from irradiation stress, and it can show to suppress DNA damage by 40% via X-rays. And so this is significant in this study, and you can see here in the orange, the D-sub treated DNA has a lot more proliferation than the normal DNA when induced with radiation. So very promising, and that's what we kind of just want to figure out and see if we can relate it back to humans. So now microbiome isolation. Isolation of the tardigrade microbiome is extremely difficult because they're so resilient. So this will include the tardigrade and the microbiome DNA when we extract its DNA. So we're going to try and utilize 4 different DNA kits and figure out which one is the best at extracting that DNA from the tardigrade. A brief summary of each kit. First one we have is the Quagan-Dean Easy Blood and Tissue Kit. It's very rapid and it does the total DNA, so it has genomic, mitochondrial, and pathogen DNA. And it usually yields around 15 to 25 micrograms worth of DNA per mil, and that's good. That's usually the range that we want, but the only downside of this kit is that it has the total DNA. So we want to try and see if we can find a kit that kind of separates it a little bit more for us, makes us do less work. Then the next kit is the Intramin DNAzole kit. It's isolation of just the genomic DNA of the tardigrade. It can do solid liquid samples, but it only gives us about 3 to 5 micrograms of DNA per mg of animal tissue, which when you're dealing with microscopic animals, that's a little difficult, especially to only have that little yield after doing the kit. So that one's not really our favorite right now. Third and fourth, we have the Introgen PureLink Microbiome DNA Purification Kit. It's very fast, and microorganisms and a variety of human samples can be used, like we want, because the tardigrades are microorganisms. And this one gives us a really wide range. It just depends on how specific your sample is. So if you have just a bunch of tardigrade tissue, then you'll get a pretty great yield, about 20 mg of yield. But if you have that tardigrade tissue mixed in with a lot of other things like lichens that they're attached to, then you're not going to get as well the yield. So that's kind of troublesome because they're microscopic, so it's very hard to separate them from their food source. And then lastly, we have the Intramin PureLink Genomic DNA Kit. This one, again, only does genomic DNA, which is a little better for our case. And this gives us about 5 to 25 mg of yield, which is similar to the last one, just depends on how pure your tissue samples are. But this one gives us very sharp banding because it can give you a higher purity and a higher concentration than all the other kits. So that once we go through all 4 of these kits, all of our samples, we're going to put it into the cubit. The cubit tells us how concentrated our samples are in the end once we have our DNA extracted. And to do genomic sequencing, we want to have about 15 to 25 mg per mil, like I said earlier. It's highly selective over DNA, of double-stranded DNA over RNA, which is good because we want DNA, not RNA. And then it's tolerant to common contaminants, such as free nucleotide solvents and proteins, which is also very good because then that means that we just have DNA in that sample, not everything else, when we go in for genomic sequencing. And so, so limitations to date. I will not be telling you all which kit we decided on because we haven't been able to actually test those kits yet. Because like a lot of research, we ran into some problems with actually finding the tardigrades. And so here I have a picture under the microscope of what a typical sample will look like from the lichens. And here I have circled a couple little brown bits, but it's a little hard to distinguish whether or not those are possibly tardigrades in the ton state, or if they're just dirt, or maybe it's bark or something from the tree. And so because they're in this ton state, it makes it very difficult to extract them. And we don't want to waste these kits and possibly just run bark through the kits and not get any samples. So that's our current limitation. We actually tried to get some samples from Missouri, a little more south, to see if warmer weather would give us some tardigrades, but that was also to no avail. And then we also just now gotten some samples from Texas, and so hopefully those will have some open to tardigrades. So our current and future research will try to get 10 tardigrades per extraction protocol, so that makes it 40 of total samples. If not more, we want more the better, right? And so we'll extract and quantify that DNA from the tardigrades, and then we'll run fish analysis to make sure those bacterial communities are present. And then we'll do genomic sequencing of the bacterial organisms in collaboration with UNC's core facilities. So then the characterization of microbial organisms consists of two different processes. Like I said, the fish analysis, which will let us know whether or not that 16S ribosomal RNA is present in the tardigrade's DNA sequences. We want to know this because 16S ribosomal RNA is common in a majority of microorganisms such as bacteria and archaea. And then once we know that they're present and we know that where they are on the tardigrade, we'll run MySeq analysis and bioinformatics, and then also PCR to try and bump up that, amplify the 16S RNA banding. And then MySeq is well established in studying taxonomies and complex microbiomes, so this will let us know exactly what bacteria and archaea or microorganisms are present in or on the tardigrade. So here are my references, and then the quick acknowledgements, College St. Mary's Science Department, and then Embry program, and then Dr. Grove as well for helping me. And then I'll take any questions.
How do you tease apart the tardigrades from the lichens?
We just do it physically. So we usually use a little scalpel or a teeny little needle, and we try to physically separate them from the lichen and just continuously separate back under the microscope, separate back under the microscope to get as close as possible. But
Have you tried like vortexing it to see if
we've not tried vortexing. Yeah, that's a
No.
really great. That's a really good concept.
Yeah, sorry.
No, that's
Sorry,
great.
Actually, the best way to illustrate what I'm thinking.
No, that's great.
If you put it in there, I wonder if you could homogenize it enough,
Yeah.
but then you could survey it faster and then tease it apart.
Yeah, absolutely. I think we'll definitely try and utilize it.
Are they actually eating the lichens or other microbes on the lichens?
It's the other microbes located on the lichens.
So they don't just need the lichens?
No, that's just what they adhere to and where they're